Reversible and irreversible cell injury. To be or not to be. The fate of a cell rests in the type of injury it encounters. A cell may adapt or fall prey to the injury. Reversible injury is damage from which a cell can recover completely if the injurious agent is eliminated. If it persists for a long time or increases in intensity, the cell reaches a point of no return and dies. This is called irreversible injury. What would you do if you had a machine that could predict the future? If we knew how to identify this point of no return, could we not intervene and prevent a cell from dying? That's exactly what we are going to talk about. If we need to rescue a cell, we need to recognize the signs of distress. They may be ultrastructural, microscopic, or changes in gross morphology. Let's start with reversible injury. The changes seen in the cell are cellular swelling and fatty change. Cellular swelling is the literal bloating of a cell. But why does this occur? The ion transporters in the membranes of the cells and organelles become dysfunctional and let the water in. As a result, organelles like the endoplasmic reticulum and mitochondria swell, and the cell itself swells, and this is reflected in the overall size, appearance, and weight of the organ. The cells develop cytoplasmic vacuoles, which represent pinched-off segments of the endoplasmic reticulum, like tiny bubbles in the cell. Hence, this pattern of non-lethal damage is also called hydropic change or vacuolar degeneration. Phospholipids from the damaged membranes aggregate in clumps within the cytoplasm and are called myelin figures, as they resemble myelin sheath of nerves. The cell membrane shows blebbing and loss of microvilli with dark eosinophilic cytoplasm due to the loss of RNA. This is a picture of normal intact renal tubular epithelium. The picture on the right shows the same cells with features of reversible injury, namely dense eosinophilic cytoplasm, cellular swelling, and blebbing. Another telltale sign of reversible injury is fatty change or steatosis. As the name suggests, it is simply the accumulation of fat in a cell. The most common organ affected is the liver, as it plays an important role in lipid metabolism. However, it can also be seen in the heart, skeletal muscle, kidney, and other organs. The causes can vary from toxins, protein, malnutrition, obesity, and diabetes mellitus, the most common being alcohol. The fat accumulates in the cytoplasm of the hepatocyte in the form of small or large vacuoles, thereby displacing the nucleus to the periphery. When the majority of hepatocytes are affected, it reflects in the overall increase in size and weight of the organ. The liver looks pale yellow, boggy, and greasy. We will be discussing this concept in further sessions. All these changes can be reversed if the injurious agent is withdrawn from the environment. If not, the cell reaches a point of no return and dies. Functional loss is followed by cell death. Ultrastructural and microscopic changes appear before gross features of death in the organ. Now let's learn some more about cell death. There are two modes, namely necrosis and apoptosis. Necrosis is cell death that occurs due to severe injury like ischemia, infection, trauma, or toxins. It is always pathological and uncontrolled. The cell dies with the breakdown of all the membranes and release of all its contents into the environment, eliciting inflammation. Specific nuclear changes seen in necrosis are pycnosis, or nuclear shrinkage, karyorrhexis or nuclear fragmentation, and karyolysis or digestion of the DNA. This is a picture of normal intact renal tubular epithelium. The picture on the right shows necrosed cells with disruption of the cell membrane, intense eosinophilia of cytoplasm, nuclear loss, pycnosis, and leakage of contents. It's like being at a crime scene of a messy murder. Apoptosis, on the other hand, can either be physiological or pathological. In the previous sessions, we spoke about how cell injury, adaptations, and death are an ongoing lifelong process. The cell death in this context
video will be talking
is
about
apoptosis.
the types of cell injury,
For example,
especially
the
we'll be
shedding
focusing and comparing reversible versus irreversible injury. So due to environmental changes or toxic chemical exposure, there could be injury of the cell. And when we talk about the types of injury, we have to think that there are specific organs which are highly susceptible to the injury, like mitochondria, ER, nucleus, and membrane. And we would be discussing the context of reversible and irreversible injury, keeping in these organelles in mind. So that there could be two consequences of injury. One, the cell try to adapt to the stress and regain its own physiology or function. Another thing is like more extreme solution. It's basically cell death. A non-functional cell would be eventually dying. So, cell injury happens due to stress. There are multiple adaptation strategies like hypertrophy, hyperplasia, atrophy, metaplasia, etc. So, all these adaptation strategy is covered in details in other video in the I button. Click on that. But anyway, when the cell undergoes injury, there could be two types of injury, reversible and irreversible. If the injury is reversible, it would be regaining its function eventually when the stressor is removed. If the injury is irreversible, then it might undergo necrosis,